So this is part 3 of chapter 2. We're going to move on to some more tools in the lab, specifically the microscope. I'm not going to spend a ton of time on this because it's fairly self-explanatory. So I will go a little bit quicker through this, but you can always rewind and rewatch, and certainly ask me questions if you have any. So kind of a generic microscope here. We have the oculars, the part that you look through. You have the rotating nose piece, the part that spins and all those lenses move around. You have the objective lenses. You have 3 or 4 of them depending on the microscope. Mechanical stage, just referred to as stage. This little metal piece back here is the stage clip. We have the iris diaphragm, which is within the condenser. I know this picture does not show that, but the condenser is kind of wrapped around the iris diaphragm here. So anytime I would ask for the identification of what this was, I would say it was the iris diaphragm / condenser. Light source right here. It's a light that shoots up the top. This is the on-off switch. This is the dimmer switch or the light intensity knob. The arm. The big knob back here is the coarse focus. The little knob in the middle is the fine focus. And then we have this knob sticking down vertically from the stage with 2 adjustments, which is referred to as the stage adjustment knobs. And these make the stage go front and back and side to side. Coarse adjustment adjusts the stage up and down in large increments. Fine focus adjusts the stage in small increments. The iris diaphragm changes the opening in which the light is shooting through. So it can be a very small opening or a very large opening. The oculars magnify everything 10 times. And then the different lenses are 4x, 10x, 40x, and 100x. 100 being the oil immersion, 40 being the high power, 10 being the low power, and the 4 being the scanning lens, which is where you should always start out on. And that information is here. Difference between a real image and a virtual image. Real image is formed by the objective. The virtual image is formed when the image is projected up through the microscope body to the plane of the eyepiece. The ocular lens forms a second image. Nice little picture there. Resolution is also known as resolving power. It's the capacity of an optical system to distinguish 2 adjacent objects or points from one another. So obviously the microscope has way more resolving power than the human eye. Just, you know, how good the resolution is, basically. Low resolution, you know, as opposed to high resolution. What is the point of the oil? When we put a drop of oil on the slide, the oil immersion lens reduces light scatter and increases resolution. So by putting that drop of oil between the lens and the slide, that reduces the light scatter that would be lost otherwise. Okay, so I'm just going to keep going here. Some of these I'm going to skip over. Types of microscopy, bright field. Most common type of light microscope that we use is going to be the bright field microscope. Forms when its image is transmitted through when light is transmitted through the specimen. Can be used for both live and unstained material and preserved materials, as well as stained materials. So that's what we usually use in the lab traditionally. Dark field, it's a bright field microscope that can be adapted. We add a special disc called a stop to the condenser. The stop blocks all the light from entering the object, the objective lens, except the peripheral light that's reflected off the sides of the specimen. So basically they use this to visualize living cells that would be distorted by drying or heat, and that can't be stained through normal methods. Phase contrast. Basically constructed to take advantage of the fact that cell structures differ in density. So it's useful for observing intracellular structures such as bacterial endospores, granules, and organelles, and so on. Fluorescence. It's a specially modified compound microscope furnished with a UV radiation source. There are the application of certain dyes that fluoresce the specimen, useful applications in diagnosing infections and pinpointing particular cellular structures. Confocal. Scanning confocal microscope overcomes the problem of cells or structures being too thick by using a laser beam of light to scan various depths. Most often used on fluorescently stained specimens, but can also be used to visualize live unstained tissues and cells. Transmission electron microscope, method of choice for viewing the detailed structure of cells and viruses. I don't see a lot of pictures of these, but you know, they like this because, like it says here, it can penetrate thick preparations. It's sectioned in extremely thin slices in order to view it. This is actually the coolest. If you look up scanning electron microscope images, there's some amazing images that you can see with these microscopes. The SEM provides some of the most dramatic and realistic images in existence. Color is actually added afterwards. The actual microscopic image is black and white. So when we prepare specimens for the microscope, depends on what we're looking at, why we're looking at it. So the condition of the specimen, is it living or dead? The aims of the examiner, so what are we trying to see? Are we trying to see the overall structure? Are we trying to ID something? Are we trying to look for movement? And also whatever microscope is available. So there's wet mounts and hanging drops. A wet mount consists of a drop or two of a culture placed on a slide, and then we put a cover slip on top. Hanging drop, basically we have concave or depression slides, and we just put the specimen in that concave depression, and sometimes they take Vaseline or some sort of sealant and cover that sample. And that is a hanging drop example. So unstained cells in a fixed smear are difficult to see. So, more times than not in the lab, we're going to stain our specimens. Staining is any procedure that applies colored chemicals or dyes to specimens. There are basic dyes and there are acidic dyes. Basically, bacteria have numerous negatively charged substances and they attract basic dyes, and acidic dyes are actually repelled by cells. So I'm not going to get too much into the negative versus positive staining. This is the important part, simple versus differential. So simple stains only require a single dye. It's a very uncomplicated procedure. Basically, you take a stain and you color everything with that stain. It causes all the cells in the smear to appear more or less the same color, regardless of the type of cell. And what this reveals is the shape, the size, and the arrangement, how they lay next to each other on that slide. So simple stains don't tell us much other than the shape, the size, and the arrangement of the cells. However, differential stains use two differently colored dyes, the primary dye or stain and the counter stain. And this allows us to distinguish different cell types or parts. This is a more complex procedure, requires additional chemical reagents to produce the desired reaction. So here's an example of simple stains. So the most popular stain that I'm aware of that we use in the lab is the Gram stain, and it consists of sequential or in order applications of crystal violet, Gram's iodine, and alcohol rinse, and a contrasting counter stain. So we usually use safranin for this, for that counter stain. So crystal violet goes on. We leave it for about a minute, depending on the video that you watch or the book that talks about it. It can range anywhere from 40 seconds to a minute. But you put that on, you let it sit, and then you rinse it with some water. Then you add the grams iodine, which is the mordant. And what the mordant does is allows that stain to stick, the original primary stain. It helps it to stick. And then that grams iodine, after about a minute, is rinsed off. And then we add an alcohol rinse, which we refer to as a decolorizer. And that stays on for less than 5 seconds. We rinse that off really quickly. And then we add the counter stain, which is safranin, and that stays on for about a minute. And then we rinse it again with water. And that is the procedure for a gram stain. So different results in the gram stain are due to differences in the structure of the cell wall and how it reacts to the series of reagents applied to the cells. Specifically, you're going to hear me talk about this more and more this semester, but the layer of peptidoglycan that is found in that cell wall is what causes a difference in the gram stain reaction. And we'll go more in depth in that coming up. The gram stain is the universal basis for bacterial classification and identification. It's a really quick turnaround, like 5 minutes, and you can have a result for the doctor that basically allows us to differentiate whatever the bacteria that's growing, it's causing the infection. We can say, okay, it's either in, the gram positive category or it's in the gram negative category. And that aids in getting us closer to a diagnosis, and also allows the doctor to start drug treatment, medications quicker with this gram stain. So we do them frequently in the lab. Another type of differential stain is the acid-fast stain. This allows us to differentiate between acid-fast and non-acid-fast bacteria. Specifically testing things like Microbacterium tuberculosis. So these bacteria have a particularly impervious cell that holds fast, tightly and tenaciously, the dye carbyl fusion when washed with acid alcohol decolorizer. We also use this acid-fast stain for other types of diagnoses or identifications of certain bacteria, fungi, and protozoa. Endospore stains, similar to acid-fast, distinguishes between endospores and vegetative cells. Very important when we identify gram-positive spore-forming members of the genus Bacillus and Clostridium. So here's some examples of differential stains. So we have some gram-negative rods and some gram-positive cocci. So the blue, purplish blue are gram-positive cocci. They look like little circles. And then the rods are rod-like. Those are pink. So those are gram-negative. And then these darker ones are gram-positive. All right. Let's see. So special stains used to emphasize cell parts that are not revealed by conventional staining methods, specifically capsular staining, used to find or see the capsule of a microbe. Flagellar staining, used to basically reveal the flagella for bacteria, or in bacteria or protozoa for that matter. So that is it for chapter 2. The rest is the appendices. And you can, this is what the capsules look like. But anyway, so that's that. If you have questions, definitely let me know.